My name is Prof Fedrizzi. I'm a professor of medicine at Federal University of Santa Catarina, Brazil. It's an honor to have been invited to participate in this event. First, I would like to thank for the invitation to participate with renowned researchers. Thank you on behalf of me, my team, and my university, where I coordinate a research center. A special thanks to Dr. Christine for the invitation. I apologize, but unfortunately I couldn't be presenting this day. When the COVID-19 pandemic started in March 2020, unlike how now infections or epidemics, our research team began to ask some questions such as, first, is there any vaccine available on the market that has any action on COVID-19? Other researchers also began to ask the same question with some scientific publication about it. For several years, a lot of studies in the field of immunology have demonstrated new knowledge about our body immune response to infections, such as beautiful works developed by our colleagues who participate in this event, Dr. Christine and Dr. Niteya. New concepts emerged as innate immune response training, innate immune response memory, and heterologous immunity extended. Our objective in the search for a vaccine already now to act against COVID-19 was precisely to evaluate in a clinical study this training of innate immunity, its memory and protection against a disease for a long time. The second question was, what are the evidences of heterologous immunity with MMR vaccine? There are a lot of papers theorizing this possible action of the MMR vaccine in heterologous protection. Some results have already been described, as in this paper by Dr. Christine and colleagues, where a decrease in the hospital admission for any infection with recent MMR vaccination was observed. The third question was, what are the evidences of the action of the MMR vaccine on COVID-19? Here we also find a large number of articles about this possible action of the MMR vaccine against COVID-19. In this paper, for example, Fidel discusses the possible action of trained innate immunity to decrease the severity of COVID-19. In this population-based study, Gold and colleagues demonstrate a possible relationship between decreased mortality from COVID-19 and a population with a high vaccination coverage with the MMR vaccine. Observational studies have demonstrated a decrease in the severity of COVID-19 in persons vaccinated with MMR vaccine. Relationship between the severity of COVID-19 and the use of MMR, BCG, and hepatitis B vaccines in a group of primary care physicians, demonstrating a significant decrease in severity in those who received the MMR vaccine. The fourth question was, what is the action of the MMR vaccine on cellular immunity? The SARS-CoV-2 virus has an inhibitory action on the interferon, which leads to an increase in viral replication. The same mechanism was described by Brodin and by Larenas Linnemann and colleagues. The MMR vaccination can stimulate cellular immunity with increased production of interferon and natural killer cells. And when we come in contact with SARS-CoV-2, the virus reactivates memory T cells generated by previous vaccination by Tdap and MMR vaccine, stronger with MMR vaccine, which may reduce disease severity, demonstrating the cell immunity memory. The fifth question was, what is the action of the MMR vaccine on humoral immunity? Protein homology between the SARS-CoV-2 and rubella virus may be protective against severe disease and death by COVID-19. The microdomains of the SARS-CoV-2 virus and rubella virus have an amino acid sequence with 29% identity. A 30 amino acid sequence is homologous among the SARS-CoV-2 spike glycoprotein, the measles virus fusion glycoproteins, and the rubella virus envelope. In this article, the same observations on the protein homology of the measles virus and SARS-CoV-2 may contribute to possible protection against COVID-19. And finally, there is significant evidence of a higher concentration of anti-mumps IgG antibodies and the lower severity of COVID-19. The sixth question was, what is the mechanism of MMR vaccine action? The concept that patients with COVID-19 who have a higher viral load and more likely to have severe illness, and the concept of training and memory of the innate immunity of our colleagues, Niteya and collaborators, could act in reducing the severity of COVID-19 by decreasing the viral load. Our body may have a potent activation of host defense leads to suppression of viral replication, which subsequently leads to low levels of inflammation, less severe symptoms, and a good prognosis. If the vaccine, unlike those who don't, would stimulate innate immunity, reducing the viral load of SARS-CoV-2, reducing the severity of the disease, and preventing the spread of the virus. In summary, some explanation that indicated that the MMR vaccine may have some action against COVID-19. Potent stimulation of the innate immune response and its memory. The MMR virus and the coronavirus are RNA virus. And 30 amino acid sequence is homologous among the SARS-CoV-2 spike glycoprotein, the measles virus fusion glycoprotein, and rubella virus envelope. The macrodomains of the SARS-CoV-2 virus and rubella virus have an amino acid sequence with 29% identity. There is significant evidence of a higher concentration of anti-mumps IgG antibodies and the lower severity of COVID-19. Heterologous adaptive immunity by reaction of MMR vaccine-induced memory T cells. Well, let's go to the result of our trial. The result of this first analysis of our study were published in the Journal of Clinical Trials in February of this year. This is a randomized, double-blind, placebo-controlled phase 3 clinical trial to evaluate the efficacy of the MMR vaccine against SARS-CoV-2 induced COVID-19 in health workers of a hospital and clinics from Florianópolis, Brazil, seen at the University Hospital at Federal University of Santa Catarina and started on July 2020. A total of 430 health workers were included in the study. 6 were excluded because they had a rapid test for SARS-CoV-2 positive. 424 were randomized to receive the MMR vaccine or placebo, with a median follow-up of 5 months. Participants who received at least one dose of vaccine or placebo were analyzed in the intention to treat efficacy analysis, and participants who received 2 doses and had no protocol deviation were evaluated in the per protocol efficacy analysis group. Volunteers with a rapid test and RT-PCR negatives at all visits were included in the efficacy against infection assessment, and volunteers who had any of these positive tests were evaluated for the severity of COVID-19. The details of the study can be seen in our publication in the Journal of Clinical Trials. In summary, we had 430 healthcare workers aged between 18 to 70 years, randomization 1.5 for MMR group and 1 for placebo group. Visits each 4 weeks, more or less a week. Visit 1 to visit 7. Rapid test positive on visit 1, exclusion of the study. Visit 1 to visit 7, rapid test and RT-PCR to SARS-CoV-2 were performed. Visit 1 and visit 3, 0.5 ml subcutaneous MMR vaccine or placebo. Extra visit when COVID-19 was suspected. Adverse events were evaluated up to 30 days after the injection. After visit 7 was realized interim analysis. RT-PCR exam for SARS-CoV-2 collected at all visits and performed at the Central Laboratory reference in Florianopolis. Rapid test IgG and IgM for SARS-CoV-2 performed on all visits by my team at the University Research Site. And what were the results? There was no significant difference in sex, age, race, comorbidities, and profession in both groups. There was no significant difference between the 2 groups regarding the use of the MMR vaccine prior to the study. There was no significant difference in sex, race, age, or profession when evaluating the case of COVID-19. When assessing comorbidities, hypertension, obesity, and neuropathy were the most frequent in both groups. However, we observed a higher risk of participants with diabetes becoming infected with SARS-CoV-2. We had 33 asymptomatic RT-PCR positive participants at the visit 1 and visit 3 who received the vaccine or placebo. In the follow-up, the participants who received the vaccine, 69% remained asymptomatic, while in the group who received placebo, this percentage was 30%. And the big question is the MMR vaccine able to reduce COVID-19 infection or severity? Among the 424 participants who received at least 1 dose of the MMR vaccine or placebo, there were 102 cases of COVID-19. with no significant difference in cases of infection between the groups. The MMR vaccination didn't prevent the SARS-CoV-2 infection. Of the positive cases in each group, we observed a significantly higher frequency of asymptomatic case in the MMR group than in the placebo group. In the intention treat population analysis, where the participants received at least one dose of the vaccine or placebo, the MMR group had a significant risk reduction of 48% for symptomatic COVID-19 and a 76% risk reduction in the need for treatment for COVID-19. In the susceptibility population analysis, where the participants received 2 doses of the vaccine or placebo and had no deviation from the protocol, the MMR group had a significant risk reduction for symptomatic COVID-19 of 51% and 78% risk reduction in treatment. There were no cases of severe COVID-19. In conclusion, this interim analysis suggests that compared with a placebo, the MMR vaccine reduced the risk of COVID-19 symptoms and reduced the severity of COVID-19. But our study had some limitations. One, number of the participants was small. The median follow-up was 5 months. The duration of the action of the MMR vaccine in COVID-19 disease progression is unknown. We don't know the cellular immune response and the viral load between the vaccines and placebo. Genetic sequence of suspected cases of reinfection was not performed. The dosages of antibodies in the individuals who developed COVID-19 were not performed. The genotyping of SARS-CoV-2 variants were not performed due to laboratory challenges. The age range evaluated was 18 to 60 years. Results may differ in other age groups. The participants were health workers, and it is not possible to extrapolate the same result to the general population. Finally, the last question is, what is the results of our trial? Well, the MMR vaccination may be a potential strategy to reduce severity and the mortality of COVID-19. MMR vaccine would be useful in several populations in the world that don't have access to the COVID-19 vaccine made in the African countries. For example, in these countries where the coverage of COVID-19 vaccination are extremely low. And the second applicability would be in a new epidemic or pandemic as an emergency measure until specific treatments or vaccines for each case are available to the general population. Again, I would like to thank for the invitation and apologize for not being able to attend this webinar. Here's my email for any contact. Thank you very much.